This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Be aware of that. There's companies, you pay them $50 and they will run your name and image through all kinds of databases, and they will actually give you a profile of what they find on you. So they can work up an entire personnel profile. This is what ASIO does anyways, if you ever apply there. But I did it once, it cost me $55, and it is freaking terrifying how much information, personal information about you is floating over there into the ether of the internet, and how much someone can gather together and put in a file for you. So I got a 40-page document on everything I'd ever done in my life, on my political inclinations, my causes, my whatever, education, my papers, my non-attributed work, my personal preferences. It was terrifying. So anyways, if you have $50 and you're curious, do it. But I am a firm believer in not having a Facebook page, even though I have one for my relos from overseas. Otherwise, they Skype me at 4 in the morning because they haven't worked out how time differences work. Anyhow, have a look at your Facebook. Either set it to private or remove those photos that aren't, kosher for employers. And try not to, I know it sounds terrible, but try and remove all those posts where you're supporting all of these questionable or not so questionable political causes and that sort of stuff. So, or just delete it all or set it all to private, whatever you do. Make sure you Google yourself and see what you find. Okay, I forgot to mention MedTech Has Talents and MedTech Actuator. They're really good. They're specific for medical devices. I think I've done one of their things in the past. They're good for startups because they're national and they have a lot of funding, and they actually invest seed funding, which means the initial stages of so if you're successful, if you go through their process, they will give you up to $200,000. And they do take some equity, but it's fairly reasonable. I think it's like 1% for $10,000. So if you want $50,000, 5% is quite reasonable, I think. And they've been known to be quite helpful. So I just thought I'd mention them. But usually if you want to get through to the actuator, you need a prototype. But it's just to be aware of. And please, please, please fill out the subject experience survey. It's open until the 9th of June. Someone told me it was closed. It's not closed. I checked. They really rely on you guys to get feedback, I don't know if you guys know this, but the reason I'm here is because of the survey. So to get people from industry here to talk to you guys, you need to actually request it. You need to give feedback. I'm not fishing for compliments. I'm going to be long gone before these results ever come out. But I want you guys to tell them what worked, what didn't work. Usually what happens is when people don't like something, that's when they complain. But they don't tell you if they're happy with what they've received. Generally in surveys, it's more of a negative thing. So if something worked really well for you, if you enjoyed something, I know that having the career talk was not part of the curriculum. I just added that in for you guys as a special thing at the end of the workshop. If you thought it was helpful, if you enjoyed it, put it, request it for next time. And if you thought something was way too hard or you didn't have the appropriate background for it, and it was covered in the subject and you're expected to know it, and it was just inappropriate, complain. Right? Not just for this subject, for it to carry value. So there's, as I said, there's power in having data. For it to carry value, more than 50% need to complete the survey. More than 50% of students, and I know that at the moment they're not anywhere near that. So please, 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 it takes 5 minutes. Just sit down and have a whinge or whatever you want to do, and be honest with it. Okay, final exam. I know you guys are all going to do really well. I'm so proud of you. It's super exciting. Now, as I said, there's 2 exams. There's the main exam and the supplementary exam, and I can't exactly remember which one's going to be which, so we'll just cover everything. It's a 3-hour exam worth 60% of your end-of-year mark, and it is a barrier exam. You have to actually pass the exam to pass the subject. Okay, so you get 15 minutes reading and 180 minutes of writing, and hopefully Lee's going to pop in at some point today to talk about her section of the exam. So it's worth 120 marks. Out of those 40%, 40 marks are going to be based on Lee's content, and 80 are going to be based on what I've taught you so far. So, yes. So 33.333% Lee, and the rest of it is all me. Okay. So we have part A. There's 4 questions, and they're very, very similar to what you had in your midterm. And Lee's going to talk more about them when she comes in, hopefully soon. Part B is going to be 15 multiple choice questions for 15 marks. I'm hoping they're straightforward, but we have to make some of them hard. So it's very standard MCQs. There's probably going to be at least one answer that's absolutely ridiculous and that you can discount straight away, and then you might need to have a think about the other two or three. As a general rule, you shouldn't be spending more than 2 to 3 minutes per mark. So everyone has a different strategy to approaching exams. I personally, during the reading, I go through and I pick out the questions that are worth the most marks, and I do those first. So I'll usually do the essay question first, but I'll set myself a time limit, and I'll say, when it gets to the 1 hour mark or whatever I've calculated for this question, I put the pen down and move on to something else. Some people like to do the MCQs first, but the issue of that is if you get stuck on something and you spend too long, you turn around and an hour is gone and you've only gotten 15 marks. So it's like cost-benefit ratio. And with the MCQ, I'm giving you bad advice, but I'm like, if you leave it till the end, you don't have time to read it, you can just circle something. Generally, people just go circle C. I'm aware of this, so I haven't put many C answers, just so you guys know. But I used to do that. I used to just go C, 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 C when I ran out of time at the end. All right. So I want you guys to focus heavily on stuff we've covered in workshops. So if something, let's say a table or a flowchart diagram or an idea or an interpretation, if we've covered it in both lectures and workshops, it means you probably need to know that. All right. So as I said, I've probably weighed more heavily on the workshops than the lectures because that's the application of that knowledge, and I think it's quite important. And and obviously, if you look at I didn't put them in, the learning outcomes for this unit, you will see that clinical trial design is one of them, regulation of medical devices, and getting to market is another one of them. So we actually need to know this stuff. So we've gone through this entire life cycle by now at some shape or form, and we've spent a lot of time on session stages and not so much on other stages. So I want you guys to obviously be familiar with it. It's popped in every lecture since we started. So yes, what do you guys want to cover? Do you have any questions? I've got all the lectures here. We can go to a specific lecture. I think someone emailed me saying that they really struggled with this lecture because they didn't know it was very content heavy. That's on me, but it's actually really hard. You get given a certain amount of stuff to cover, and it's really hard to cover it with only superficial knowledge. So I will never ask you something that hasn't been, this is why I didn't give you so much reading material. I will never ask you something that hasn't been covered in the lecture notes, all right? I will never ask you to identify a device that we haven't either discussed or it hasn't been in one of those little tests we put together or something like that. Right? So, yes?
Just a question with all the medical devices that you taught us, whether they are medical devices or not, do we also have to categorize them into the classes?
How do I answer this politically? Okay.
I know you gave examples for which classes are they for only some medical devices, but there's a huge more amount of medical devices that we haven't categorized.
That's right. So I'm more interested in, is it a medical device or is it a biological or is it a medicine? You need to know that, right? I am really big on definitions for specific terminology because I think it's really, really important. Now, I know that the medical device definition is huge, so I'm not going to ask you to reproduce it word for word, but I need you to understand it. So in terms of classification, It is important, so I need you to, so it's linked to risk. So if something is super low risk, you know it's not going to be a class 3 medical device, right? So like a tongue depressor, we know that a syringe is a class 2. So we were talking about, does it touch the skin? Does it touch a mucosal membrane? Does it form, does it go through the skin and into a vein? And finally, is it implanted and left in the body? And that gives you a pretty good idea of where it stands. What I want you to remember, so if I ask you to classify something, you will get that classification chart that we used in the workshop, and then you can follow it. And if it's not going to be like a combination device, it's going to be something very unambiguous. However, I do want you to know those 3 special rules where I said if it's any of these things, I don't care what it is, if it's a Band-Aid, it's automatically going to be a class 3 medical device. And what are those rules? Is that if it has a medicinal substance, so if it has a medicine, you can have a Band-Aid that has an antibiotic ointment on it, and then it's automatically a class 3 medical device. If it contains an antibiotic, like IVF media, is automatically a class 3 medical device, and we've mentioned that like 5 times. If it has a, so something of bacterial or, oh my God, I've lost the plot. Anyhow, you get the picture. So the special rules, and there's only a few of those. Animal origin material will be a class 3, and sometimes if it has a human origin material, it might be a biological or a medical device, or it might be exempt, right? So stem cells is a typical example where it can be either a biological or it can be exempt, so not deemed to be a therapeutic good. And who remembers the difference? Come on, guys. When is a human stem cell a biological and when is it not? Yes?
It depends on if we process it or not.
That's exactly it. So autologous stem cells, the ones where you're taking out from a patient, putting them back into the same patient, is generally exempt. So it's deemed not to be a therapeutic good. Commercial stem cell lines, such as the ones used for breast cancer research or for whatever reason, will always be a biological. Okay? And organ transplants are not a biological, I think. Anyways, review those tests that we've done. Was the moral of that story. I will get back to someone emailed me about. They were struggling with, where is this lecture? Lifestyle for prototyping and design. I think it was this one, I think. All right, so we talked about valves, and we talked about syringes, and so, but what I want to get to is, so yes, imaging agents are not a medical device. I think we covered that like 3 times. Okay. Sorry, guys. Okay. Just really basic stuff, like, I really hope that you guys know this, but I'm like, who regulates what? Who regulates clinical practice and who regulates therapeutic goods? Do you guys remember? That was covered in the first lecture. Yes, and who regulates TGA? Thank you. Like, really basic stuff. I'm hoping you know that. And because we talked about, this is not the right lecture, but because we talked, so I'm not going to ask you to like spit out all the tests. I'm not going to ask you to be able to name all the standards that we've covered. If I've said you need to know the name of this standard, and there's only like 3 of those, then you probably need to be familiar with the name of that standard. Sorry, this is the wrong lecture. So we were talking about good clinical practice versus good laboratory practice. And then we're talking about all those different, I think it might be, here it is. Okay. Yeah, so I expect you to know the 2 schemes. What they are, when you'd use which one, what are the differences. They apply to both devices, biologicals, and medicines. So they apply to anything. I want you guys to know the types of clinical trials. So the ones that we covered, the big main ones. I don't want you to know like that really random, small, retrospective, complicated crossover, just the basics. And why are they important? Do you remember how we looked at that table? Oh, it's in here. Let's look at that table. Hi, Lee.
Hi, Tim.
Welcome back. I hope you're feeling better.
Hello, everyone.
I've already talked about the outline of the exam, so I will...
You showed them the front page?
No.
Okay.
Are we allowed to do that? I didn't think we were allowed to do that. Okay, cool. But I
You finish what you're doing anymore.
all right, now, there was just one question. Someone said, out of all this stuff, what do I need to know? So I wrote this about the ethics when I thought you guys knew basic ethics. So we've gone back to the drawing board in one of our workshops, so I want you to know basic ethics. Cool? Yeah. You know, there's 5 principles and how they apply to when you're designing your clinical trial. The do no harm, do good, do this, do that. You get the gist of it. It's all right. So I don't want you to be able to name all the acts. I just want you to know that the therapeutic goods regulates how the TGA regulates, and there's a specific act that regulates how we deal with medical devices. I want you to know the schemes. Don't worry about this. This was just a nice little background thing about where, how basic ethics tie in with clinical trial design, but we went back and we covered basic ethics, so don't worry about the slide. So some people were saying, if it's super text heavy like this, how much do I need to know? Right. So there's only a couple of slides that are this bad, and that's because I didn't want you guys going off and reading this entire statement. I need you just to be aware of the name and what it is, right? And how it's relevant to clinical trials. I'm not going to ask you anything that isn't on this slide. So same with good clinical practice. We've covered this 5 different times. It's important. Someone was saying, oh, out of all the GCPs, GLPs, all the rest of it, which ones do I actually need to know? Well, the 2 most relevant to us are good laboratory practice and good clinical practice. I'm not going to ask you for all the principles, but I need you to know what it actually is and why it's important when we're designing a clinical trial. Right. So these were the ones I got emailed about. So it sort of goes through, we went through this in the lecture, so I'll just leave it. Any other questions? Yes?
I'm still a bit confused about the difference between CTN and CTX.
Okay, let's look at it. I think I put a table. Okay, here we go. So one's a notification, one's an exemption scheme. Right. And one is you just tell the TGA that you're doing this clinical trial with this device, and you say, look, it's super safe, it's been used overseas, it's just a new iteration of an existing device that I want to trial in this country. Is that kosher? Is that cool? You just fill out a form and off you go. And you don't even need to wait for acknowledgement before you actually start doing your trial. The other one is, this is brand new, I want to actually put it into people, I've done some testing, but it's never been done before. Can I get permission? So you're actually asking for permission, you send them your data, the TGA actually assesses your data. So this is separate from what you would have done to get ethical approval. So in addition to your ethics approval, once you've gotten your ethics approval, you submit that to the TGA, right? And they evaluate it. And you have to pay a fee, and then you have to actually wait for them to give you permission before you can start the trial. So one is just a tick this box, fill out a form. The other one is an entire process. And you look at the risk, and that determines what you do when, right? So anything that's generally high risk and doesn't have a predicate device will fall under this scheme. So anything that you guys developed, with the exception of one device during your workshops, will fall under this scheme. So I just want you to know the difference between them. I will show you. So yeah, we talked about when is it appropriate to do what. Just revise it, and here we go. Is that clear? Yes?
The CPN leaves a problem where people can just use the device and notify them, even though it probably doesn't fall under CTN, but it's not being assessed before they use the device.
But you have a responsibility to make sure that it actually does. And some of them do get audited and can get in a lot of trouble. So generally, if you're If you're not sure, then it's always CTX. If you have to even ask about it, then it's always CTX. And you can submit CTX and they can turn around to you and say, oh, actually CTM was more appropriate for you, and that's perfectly fine. They do that sometimes. But as I was saying, I provided examples of which one. I'm not going to go through all of it because we don't have a lot of time, and there was a lot of questions that came through. But yes, clinical trials, and we looked at this table several times, and we looked at this table several times. So might be worth, and we actually applied this table during one of our workshops. So always go back to the workshops and see how this knowledge is applied and make sure you understand that. I know that we did it as a group, but hopefully you didn't, you didn't let your teammates do all the work. All right. Any other questions? Come on, guys, this is the time. Is there a particular lecture that you want me to go through again? Yes?
The different types of bias, could you please explain confounding bias again?
Let me get this question.
The example we had was of like coffee and lung cancer or something.
Yeah, probably wasn't the best example. So I do want you to know the different types of bias, but I also want you to know error and how bias ties in with error. Do you teach this, Lee? Did you?
Yeah.
Yeah. Oh, okay.
From a statistical point of view.
Well, okay, let me find the relevant. This probably wasn't the best lecture. No, it was amazing. What am I saying? Case study. Oh, I know that there's not as much emphasis on the later lectures, but I do, there are questions that were done after the exam was already written, but there are questions in the exam from the later lectures as well. So like especially yesterday's lecture where we talked about registries. So don't worry about the ethics of hype, but do worry about the registries component of it. All right. Medical regulation, design, and evaluation of, oh, that looks relevant. Let's have a look. Okay. Oh. So I want you to know why are medical devices different from medicines when it comes to designing your trial. Okay, where's the bias? Sorry guys, I'm getting there, You sure? Oh, my bad. Okay. Here we go. Bias and error. So this is, next slide. All right. Oh, okay. Yes. So this is when something completely different still has a relationship with your outcome. So I was just saying this, I probably didn't use this example. Coffee drinking and lung cancer. We know that smoking is associated with lung cancer. However, we know that if you looked at specifically coffee drinking and lung cancer, you would find that coffee drinking is in fact associated with lung cancer. However, if you broke it down, you would realize that most people, back when this was done, actually smoked with their coffee. So even though coffee is not directly related to lung cancer, if you looked at statistics alone, there would be like an undeniable association with it. So, I don't know, am I explaining that properly? So it's something that is related to what you're measuring, but it's not directly associated with it. So you can get this false measurement. So, and you go, but why? So yeah, so it's just about understanding what it is you're measuring. And one of the things that we look at when we design a clinical trial is we look at possible sources of error. So going back to error, and we always account for selection bias, information bias, and confounding factors. So you can't always control the confounding factors, but you try and identify as many as you possibly can. So for example, a little old lady with blood pressure probably also has diabetes, right? And if we're looking at renal function associated with one certain type of drug, we might find that there is an association. However, it might just be because those patients are more likely to have diabetes, which causes reduced renal function rather than the drug itself. So that's another example. Okay, we talked about errors. So I want you to know the difference between random error and systematic error. And I probably didn't explain this. Oh. Okay, any other questions? How does a device get to market? I've given you lots of, I haven't actually given you a lot of reading. I've given you just a couple of papers, like summary papers to look at. Maybe give those a quiz before the exam. And I do want to just look at some of the workshops that we did. Okay, where are the workshops? Workshop 3, what did we do for that one? Oh yes, basics of medical ethics. We talked about these and how they apply to how do they apply to the questions that you need to ask when you're designing your study for your device. We were talking about risk versus benefit, and we spent a lot of time on risk assessment. Please, please, please have another look at that lecture. Be familiar with the terminology and be able to apply it just in soft terms. Okay, so principles of risk assessment. Okay, what else did we do? The difference between preclinical and clinical testing. I don't expect you to know all of this. Just be aware that there's a relevant standard for whatever testing you do. And this is one of the standards I wanted you guys to be able to either name or be able to recognize. And we spend a lot of time discussing that. Okay. And we talked about how risk is based on the category. So risk determines the category of your device. Does it just touch the skin? Does it touch a mucosal membrane? Is it implanted into the body and left there? Or do you just use it as a connection, such as a needle or a catheter or that sort of stuff? So that dictates risk, as well as how long the patient's exposed to it. So a permanent device would be much higher risk. That's something that you only use for like half an hour while you draw blood, or you leave it in there for a couple of days, right? Okay. Any other questions? Yes.
So in the workshop, one of the questions was name a severe risk of your specific device.
Yes.
But in the category or in the table, severe risk is a definition with certain consequences, but if the device itself doesn't have any risks that are that severe,
so the classification of risk in that manner was only relevant if you're doing a risk assessment and you're doing a risk minimization strategy. The classification of risk in general, when you're just identifying the risks, is based on the likelihood of failure. So you know how we said about you can have a high likelihood of failure, but a low risk outcome. So something super common like your Band-Aid just falling off, but the outcome is, oh, maybe it gets infected. And then you have like a very low likelihood of failure where you have a catastrophic outcome where the stent gets loose and moves in your circulation. When we talk about identify a risk for a particular medical device, unless I've stated for what purpose it is, you can say that a high risk means that something that's highly likely to go wrong. There's always going to be some level of risk associated with a device. There's nothing is risk-free. We always talked about the fact that you're always going to have that one person who's simply allergic to what you put in their body, and they're probably going to have a reaction and die. So there's always going to be a low incidence of severe risk. But what I mean is identify the risks why your device is most likely to fail. Right.
I guess my question is the frequency of the risk defined how severe it is, or the severity?
If the question asks you that, I will specify it. So if I want you to talk about the frequency of risk, it'll say that in the question. It'll say, please identify high frequency or low frequency and that sort of stuff. But I think I've sort of trying to remember, you will need to be able to identify risks associated with a particular type of device, and you might just need to comment whether it's deemed to be a high or a low risk. So, you know how we talked about anything that has a medicinal substance, anything that's permanently inserted into the body, anything that uses an animal origin device, anything that is used in the central nervous system or the vascular system, if it's used in the heart, it's automatically high risk. So, yeah. Okay. It's a lot easier in a lecture than it is like this, but I didn't prepare particular questions because I felt like I've given you guys enough, probably too many hints throughout the study. So if you, when you're...
From my point of view, sitting here listening to this, you've been given some major hints.
You have, to the point where it's not, I'm going to get in trouble if Lee sits down and listens to all the lecture notes. But
But I know say that having no recollection of what is on band and half of the exams. I don't think that I'm giving you a hint by saying that's some major hints. Just from the way that Sam has been talking, if I'm here and I'm going, oh. So if you might, I think you've given lots of...
okay, so I just really want him to do well. I want everyone to do incredibly well and be super successful and do amazingly well. And I'm sure you will. And yeah.
Shall we talk about the exam front page then?
Yes, now I could, do you have it? I'll show you what I covered for them already.
No, I just, have you got, if you open up a web browser, and because I put it up on LMS already.
Oh, okay. So I covered this bit.
So for anyone that's doing medical imaging, I've just realised I forgot to make the announcement about the lab visit today, so I'm just sending the email now. I mean, in lectures we've talked about it, but
There's a lab visit.
there is.
Okay.
All
All right, let's do this thing. So, can we?
Yeah, absolutely.
I'll let you do that because this computer does not like me.
So just very quickly while I'm loading this, the statistics part, so my part of the exam, if you want to know how to study for it, the questions will be of a very similar format and difficulty level as the mid-semester test. That's the plan, all right? So there should be no surprises there as to the type of questions that you're going to get. All right, so on LMS, it's an engineering policy that we show you the front page of the exams, which is some nice hand holding. So under documents on the LMS page, there's the 2019 front page of the exam, and of course this is every academic's worst nightmare that they accidentally put the exam file up. So, but I'm pretty sure this is just
That's
the
why
first
I
page.
wasn't like touching it, and that's why my computer's not plugged into this because I know I'll accidentally scroll over it and you guys will see everything.
So look, here is no, it's good. It's the first page. So I would recommend that you have a read of that. But let's just quickly go through it. Okay, so yes, you can bring in your FX 100 calculators if you wish. They're going to be,
You don't.
and that my understanding is that all the FX 100s allow you to do as distinct to the 82s is some complex numbers, which for this subject won't get you very far. Magnitudes in phase? No, I don't think so. All right, so we've written it. There's a total of 120 marks. So you've got 180 minutes, 120 marks. You work it out. But here is the complicated part. There is 3 parts to this exam. So there's part A, 40 marks, my section. You will write that in one script book. Okay. Part B, that's the multiple choice.
MCQs.
Yes.
15 MCQs.
So 15 marks for a multiple choice where you will be given a multiple choice answer page, right? And then part C is Tarynka's.
No, I think they do it on the booklet. So for the MCQs, you actually circle it on the
Oh,
booklet.
sorry, I'm getting my subjects mixed up. Yeah,
That's all right.
yeah. Our first years we do on an answer sheet. Yep. Yes, sorry.
So yeah, you just circle it on the booklet. If you change your mind, make sure you cross it over really, really well and then circle the other one.
So
But
you
try
will-
not to change your
yeah.
mind. Usually your first instinct is the most correct one. I've been burnt so many times in the past where I've like crossed out the wrong answer after I've had 10 minutes to think about it. So resist the urge to change your answer. Go with your gut.
So your exam booklet is going to be marked in your script booklets. And I say plural because there is a part C,
Yep.
which is the actual written part of Tarynka's part.
Yep, and dot points are totally fine. If you can draw a diagram for it, do so. just make sure if it asks you to apply or interpret something, I don't want you to just spit out what you think is the relevant information. So if I've asked you to apply, explain to me, or if it says explain or apply or whatever, explain the difference between CTX and CTN, or if explain the difference between a biological and a medical device, I don't want you to guys just spit out the two definitions side by side. I want you to actually be able to explain, right? So just link them together somehow. Yeah. Yes, so yeah, read the question 3 times. Don't just... The exam questions are designed so that you can get some marks just by regurgitating stuff that you've absolutely memorized and not understood anything in the world, because we want people to... We want that bell-shaped curve, right? But to do well in this exam, you actually need to be able to understand how to apply some of those principles. If you can't do it on every question, that's fine. Do the best that you can. But I promise you, if you just use common sense, even if you don't know anything, you should still be able to do really well. Like, if you look at an MCQ, one of them is clearly going to be wrong, right? Then you're going to have 2 that are semi-wrong, and there's going to be 1 that's correct, right? And even if you don't understand the terminology, you know how I was saying, what's the difference between validation and, what was the other one, verification? If you can't remember the definition, but you know that one involves, did I build the right device, or did I build this device right? So those were the 2 differences. Even if you just put that, you'll probably get half the marks for the definition. So, you know, if you can't remember, do the best that you can.
Just on the bell-shaped curve thing, I think students often have a question about whether we grade to a curve or not, and I can tell you now, no, we don't. The whole idea is that we are meant to set exams such that the outcome has a nice distribution. Right? We do not take the results and go, oh my goodness, that's meant to look like a normal curve, all right? So if you get 95 out of 100, or whatever it is, 110 out of 120, you can be guaranteed that you are not going to be brought down if everyone else got 110 out of 120. All right?
I think that we're hoping for the other way.
The other way. Yeah, no. Any questions? Any of that?
And I promise you that the supplementary exam is not easier than this one. So they're both the same, because I wrote it as one exam, and then Lee went, oh, that's too much, so we just cut it into 2. So.
Yeah, you're lucky that Brooke and I had a look at that exam. You guys were going to be sitting there for hours.
I got really enthusiastic about writing exam questions, and I was like writing them, I was going, oh, I'm like, oh, they need to know this, and they need to know this, and then Lee looked at it, and she was like, so she was like, yeah, we need to not do that.
There's an art to recognising how much you can get done in 3 hours.
So everyone should be able to get like really good marks for my section of it. So, you know, everyone should be able to pass.
All right. Anything else we need to cover? Is that a question, Conchita?
Yes.
You mentioned a few times about being like an essay question, something like how many marks we're talking about. Is it?
Okay, so there's no such thing, there's not an essay question as such, but there is a question that has a sequence. So it's one topic, but it might have 5 questions associated with it. Please always just look at the number of marks associated with each part of the question. So, for example, I'm just trying to think of one where I deleted the question.
Well, let me ask it a slightly different way, maybe. Is there any part of the exam where you would be wanting an answer that takes up a full page in a script booklet?
Yes.
No, I think she meant there's one question that's broken into 5 parts.
Yeah.
And about 5 parts might take up a page.
But for each...
Yes.
I mean, for part A of that question, would you be expecting a full page?
No. So if the question says, look, I said to you, don't you expect to spend... Oh, God. You're giving me a heart attack. I'm like, did I do the right thing? Okay, have a look at the marks. If the question is worth 6 marks, multiply that by 2, so you'll only be expected to spend about 12 to 15 minutes on it. So for each mark, no more than 2 to 3 minutes per mark. If you're spending 5 minutes on a question that's only worth 2 marks, then you don't spend any more than that. So if you've gotten to your 5-minute mark, leave it, go to the next one. You don't want to spend half a page writing an answer for a question that's only worth 1 mark and then run out of time for the question that's worth 20 marks. So there are questions that are worth 10 marks, There are questions that are worth 16 marks in several parts, even if you haven't answered the question above it. So if we, you know how we said there's a series of questions, and you will find that one question will say name this, and the other one will say explain how what you named above is relevant to X, Y, and Z, right? So that's the one that's going to be worth lots of marks, whereas the name this question is going to be worth less. Even if you haven't named it, try and make something up for the following question. So you can still get full marks for the question below it, even if you haven't answered the one above it. So please, please, please attempt all parts of the questions. Okay, there's not going to be one that says choose between this and this question. You have to answer everything that's in that booklet, or at least attempt to. I am totally fine with you guys using diagrams to explain something, but please make them clear and readable, because if I can't read it, I'm going to not give you the marks for it. So if you're using shorthand, you've got to explain the definition of what you're putting in there. So I like definitions. I've included them in every lecture. So if you're going to use shorthand, if you're going to say TGA, okay, I know what the TGA is. That one, don't worry about it. But I'm like trying to think of a good one. NJR, the Australian National Joint Registry. If you're going to use that as an example for whatever you're talking about, I want you to actually be able to name it. Don't just say NJR. Because this needs to be understandable and marked by someone that doesn't have any clue as to what the material is. Right? And if the answer marking criteria says they need to name this term, if you use the shorthand, you might not get any points, or you might only get half a point. So I'm not sure if I'm going to be the person marking your exam or someone else. I think it's going to be me.
It's not going to be me, I can tell you.
But for, you know, if there's ever an argument as to what the answer was, for you to be able to get those marks, it needs to match up with what the answer criteria is specifically. So yes, use your terminology appropriately, use diagrams, don't write an essay. Dot points is completely fine.
Yeah.
Dot point the hell out of that.
There
I'm
is
totally fine
actually
with that.
nothing worse than marking a short answer where somebody doesn't know the answer and they've waffled.
Yep, and
And
you
they just go,
keep
oh,
trying
well,
to
it might be
find
this, and
something
then
to
it
get
might
a
be
marks
that,
for.
and you'll end up with the examiner's head exploding, and it wouldn't necessarily be to your benefit, right? Everyone tries to mark absolutely objectively, not subjectively, but it's human nature that if you have hidden an answer in some scrawl that covers a couple of pages, there's much more chance that the examiner is going to miss the fact that you've actually answered it correctly. Having said this, I can imagine now that there will be 62 people wanting to come and see their exam, which you are completely at liberties to do. Okay, so it isn't that we are just going to go, they've written too much, I'm not even going to read it. That's not the case. But obviously try to keep your answers. A short, elegant answer is better than a long, waffly one.
And just make sure you've answered the question. As long as you've answered the question, resist the urge to actually fill it in with more waffle.
Yeah, exactly.
I know that people think if I just keep writing, statistically speaking, I'm bound to stumble upon the correct answer. I'm guilty of this myself. I've written like booklets and stuff. But chances are, look at the cost-benefit. The time you're going to spend writing that, hoping that you'll get that one extra mark, you're going to miss out on 5 marks elsewhere. So just like move on. If you don't know the answer, do the best you can and move on.
Okay, good.
Yes.
I guess this is part of like kind of into what you said, but with like describing questions or short answer sessions, like sometimes in exams where you have more time, you can just list like more points than the marks are worth.
Go for it.
And
I don't think you should think 1 dot point 1 mark.
Oh, no,
that's
no.
a very dangerous thing to try and do.
But sometimes it's like other ways, so it's like 4 marks and I have more like extra time and I list 10 things and expecting like 5 of them probably like not relevant, but just one of the best. Like, do you
I'm not going to penalize you for incorrect answers,
yeah.
okay? If you're not sure and you want to list everything that pops into your head, do that, but you're only going to get a mark for the correct.
Yeah, I
shouldn't
reckon
say
you're
that.
playing with fire there, because
Yeah.
if you were asked to explain something and you gave a correct answer and
then an incorrect answer, you're gonna-
and hoped for the best, I know, you would lose marks because you're showing that you don't understand which the correct answer is.
And you can't apply it. So pick the best.
So it's very different between correct answers and irrelevant answers to correct answer and wrong answer, right? Yeah.
If something says describe to be the process of, and it's totally fine for you to use a flowchart or dot points. And if you want to add extra dot points, you know how we said sometimes it might get sent to a committee, which is not a part of the normal process. If you want to include that, that's fine. That's amazing. It means you actually understand what you're writing about. Or if something says please list me 5 types of registry, or something like that. I know we haven't covered 5 different types of registries, but just as an example, and you list 10, that's fine. But if you look at the question, it says, please list 4 types of registries, and the question is worth 2 points, right? So you're going to get half a point for each one. If you list 10, and 5 of those are correct, then you will get full points. If you list 10, but none of them are correct, then you're not going to get any points. Yeah.
Good.
So yes, it's not 1 answer per point. This is being recorded. I meant to turn that off.
No, this is good.
Oh, okay.
There's less than 62 people in the
Now
class.
they have it all on file.
And will we be having any review sessions before the exam?
Does anyone want a review session before the exam? Okay.
We'll put and by review session I mean consultation hours.
Consultation. Well, I've been available every week for an hour for you guys, and no one's ever booked anything with me, so I'm like, I'm either that good or that bad.
That may change, right?
Okay. Okay, because I've kind of figured out, I'm finished, and I've filled up my schedule with actual work. But
Yeah.
I am always here for you guys, so as long as you're flexible with time, I will be on site. Email, and we'll book a time, and you can come and grill me. But I will not be giving you any more hints that I've already given, and I'm going to give my word on that. So no one who comes to see me is going to get any special advice for the exam, but you might get special explanation on whatever you're stuck on.
A little bit different for the statistics part. If you come and work through solutions and you show them to me, then you probably will get a benefit out of that. God, I've just increased my
Workload.
No, it's all good. No, it makes sense. You want to work through something and you want a proper, detailed explanation, you're more than welcome to come and talk through that with me.
What can I offer? I feel like
No,
we've covered
all good.
everything.
All
But
good. All right, it's nearly time to finish anyway, so I'm going to turn the
Yep.
recording off.
Okay.
And thanks.
if you're stuck on anything, you want some hand holding, come see me.
No.